While the popular science market for longevity books is saturated, no one needs another celebrity's viewpoint on aging or another diet book, and several excellent introductory books already exist. At least a few people might want to have a molecular explanation of the exciting work that has been done in this arena. As I will explain, we have found out a lot in the past 2 decades about how longevity is regulated, which can give us clues about how we might slow aging. We now have a better grasp of the genetic pathways and cellular processes that communicate from one cell to another how to tune the rate of aging. And we also better understand the reasons that longevity is regulated at all. These insights have then led to ideas about how to slow age-related decline, and we have some good candidates for those medicines now. Some of this excitement has recently been turned into serious biotech development, with many companies focused on longevity and aging springing up in the past few years. I have been lucky enough to be right in the middle of things since 2000, since new genes that control longevity had just been revealed. The millennium was a real turning point. After bacteria and yeast, C. elegans was the first multicellular organism whose genome was sequenced, and Drosophila quickly followed. Those large-scale projects were a direct benefit of the approaches developed for the Human Genome Project and allowed biologists to carry out experiments that had not been previously possible on a genome-wide scale. RNA interference, RNAi, a mechanism that causes the messenger RNA, mRNA, of a gene of interest to be degraded, was first described in detail by Craig Mello and Andrew Fire in C. elegans in 1998, and it was quickly employed by the worm field to test all of the genes in the genome for every characteristic of interest, including aging, through new tools to easily knock down gene expression levels. This ability to rapidly test many genes in worms quickly led to an explosion of functional genomics, that is, testing of all genes in a genome for a particular activity, and the field has been expanding in many directions ever since. I got into the aging field because I was fascinated with the question of how longevity and aging are controlled genetically and biochemically. The tools that were newly available at the time, genomic expression microarrays and RNAi, allowed a previously unimaginable ability to probe long-lived mutants, that is, animals with changes to their DNA that affect a gene, and to learn what was going on inside them. The existence of complete genome sequences for all of these organisms also ushered in new genomic approaches, such as DNA microarrays, and later next-generation sequencing, allowing the analysis of every gene simultaneously and giving us unprecedented insights into the inner workings of cells as they age. The amount of data available to researchers has been exploding ever since. Genetic and genomic methods have led the way in longevity research, and large-scale studies of metabolism have added to our understanding. Meanwhile, new molecular tools, particularly the gene editing tool CRISPR and stem cell approaches, offer the exciting possibility that we might even modify ourselves to achieve better health. Because of the nature of the question, understanding how aging works, the field is extremely broad. One can attack the aging question from many different viewpoints: demography, population genetics, evolution, model system genetics, molecular biology, cell biology, nutrition science, and pharmacology. All of these perspectives are helpful in understanding how aging works and whether we can slow it down. While